I'm not going to sleep already. It's far too early. Okay, good day everyone. Well, where should I start? My name's Jono. I'm a statistician. It's my second career. Originally I was a vet. I was a vet for 13 years, and after a while I thought, I've got to change. So I'm now a statistician who mainly does medical statistics. And because of that, I worked with Mark Hutchison and Giancarlo and Paul Rowland. And Mark said, can you do a workshop for the honours students about stats? So I don't know where you are in your stats. So I'm going to try and give you all the stats you're going to need in hopefully about 40 minutes. And then we can have questions and chat and talk about any questions you have. And if you're bored at that point, we can all, well, I won't drink coffee, you can go and do whatever you want to do at the time. So what I want to do is, some of this might be revision. Some of you may have seen stats before. You already just need a review of it. Some of it will be completely new. So what I want to do is I want to just give you a complete review of what stat is, what we're trying to achieve, some of the key concepts you will be introduced to. I would say the main take-home message for today is, if you're not sure, come and see me. I'm over in the Mass building. I'm one of two loud fat Mancunians. There's another loud fat Mancunian, so, but I'm the main one. But if you ask for the large skinhead for the accent, you're pretty well going to get me. But if you want to, my name's Jonathan Chuk. Just come over, I'm level 6 over there. So, all the stats breaks down into 4 things. We call them very fancy schmancy names, but basically all we ever do is we look, we guess, we decide, we predict. So by look, that's all the descriptive statistics and the plots that you'll hear about. By guess, we're talking about what we call estimation theory. So you'll hear about things like estimation theory, we'll talk about point estimates and confidence intervals. If you ever heard someone talk about a 95% confidence interval, that's basically a guess. It's a guess with a little bit of margin of error. Decide is the entirety of hypothesis testing. So if you've ever heard of p-values, test statistics, hypothesis, all we're doing there is making a decision. And finally, what you mainly use most of your stats for is for prediction. The idea is that you've got some sort of model, and you're going to use that to predict future things. So, I wanted an example, and seeing as a vet, I thought this would be a nice one. So, the first thing you want to do is you want to be able to measure animals' body temperatures. You want to know the average animal body temperature. Does the body temperature change when you look at, whether they have a disease or not disease? Is the body temperature the same for all species? So I went and grabbed a load of data for horses, cats and dogs, so I've measured the body temperature for these, and there's 2 sorts of conditions that either have a disease state or they came in for a vaccination, so we're going to assume that they're not diseased. And the questions we want to ask are, does the disease state change the body temperature? Does it make sense to change the body temperature? Is the body temperature the same for different species? Does it change at all? So I've got species, disease state, body temperature. And I think you're thinking to yourself, look, Jono, the main thing I want to take home today is, how can I make you happy? That should be the big thing you're worried about today. If you want to make a statistician happy, what you should do is give them the data in the right way. So, I have no problem with Excel. When you record your stuff, put it in Excel, whatever spreadsheet you like. If you want to keep me happy, there's some very basic rules. First of all, you'll all have a dying urge to do lots of fancy schmancy formatting in your Excel. That's fine, but you don't need it. What I want when you put your Excel spreadsheet is every column is a variable. So in this case, we've got a variable which is what animal I've measured the results on. What's its disease state? What is its body temperature? The other thing is, notice every row is an individual subject. If you always have your data like that, such that every row is a subject, every column is a variable, you'll keep statisticians happy. And they'll be much happier. Most of my time I spend cleaning up data. And I charge $200 an hour to clean up data. If you clean it yourself, you save yourself, and cleaning data can take 6 hours, you instantly save yourself $1,200 just by these rules. This is worth quite a bit. The other thing, notice the titles are just really simple. Temp, disease state, there's no spaces in there, they're not big long descriptions, etc. The reason I like it like that is I do all my stats in a program called R. I use R because it's free, it's really powerful, it works really well. And if the data is like this, I can read it straight into R and start analysing your data. So, there's our data. Let's do the first thing, which is look. So what do we mean by look? What we're trying to do, it's called descriptive statistics, but basically what I want to do is I want to look for general patterns. Mark Hutchinson talks a lot about the stupidity test, which I think is really, really good idea. People believe statistics. In fact, 66.4% of people believe statistics even if they're made up on the spot. I'm glad someone got that joke. And the thing is, stats is just a tool. The whole idea is you're trying to show something. You've got some relationship you want to show. And you're just using stats as a tool. That's all it is. It's like a word processor or your thermometer. It's just a tool. You learn to use it well, but it's not the be-all and end-all. And the problem is people believe statistics too much. If your stats doesn't agree with what you think makes sense, it's probably the stats that's wrong. Well, there's two possibilities. One, you discover something really new and interesting. Brilliant. But a lot of the times, you've got the stats wrong. So when you start looking at it, if it doesn't make sense, for example, we know in this case, disease state, most of the time an animal has a disease state, it's probably got a fever. If it's got a fever, its body temperature should increase. So if I do plots and the body temperature is not increasing, I sort of have to go, have I made a mistake? Have I entered the data in wrong? Have I analysed it wrong, etc.? In other words, did it pass the stupidity test? The reason we're looking for it is to look for relationships. You know, how does the body temperature change when I change for different species? How does it change when I go from a normal animal to a diseased animal? The other thing is to look for obvious mistakes. As soon as you start typing data in, you will type in mistakes. So for example, when I'm looking, if I look at the body temperature and I get a body temperature of 4 degrees C, I've put it in wrong. Or for some reason I decide to take the temperature of a dead horse, which normally doesn't happen. I can recognise dead horses. So I really like visualization. The first thing I'll do once I get a data set is I will just start looking at visualizing. And there isn't a be-all and end-all with plots. A lot of people will say, oh, you should always use this plot. I use lots of different plots, messing around using SPSS or Prism, whatever it is. Lots and lots of plots to look at that relationship and say, what's it telling me? So, here we've got cat, and each one of these is the individual body temperature. So for this cat, this one had about 38.1 all the way up to 40. There's no indication of disease state at the moment. I've just looked at body temperature for the cat, for the dog, for the horse. What am I looking at here? We look at 4 things. We look at location, spread, shape, and outliers. What I'm generally looking when I look at this is I go, well, you know, do cats look higher than dogs? Do dogs on average look higher than horses? So I'm looking at what we call the location, or where's the average value roughly? There's different sorts of average values. You hear the mean, the mode, the median. But all they're basically saying is what's the average value? Around what point do you think all these points lie? The other thing I look is at spread. You know, if this is the minimum point, and that's the maximum point, we've got, you know, so much spread. How does the spread change as they go between cat to dog to horse? The other thing I'll be looking out for is there any point that sticks out? You know, if I got a point right down here with a horse, I'd be worried that I've either entered the data wrong, or it's something of what we call an outlier. It's a point that sticks out, but it doesn't make sense. And again, there's 2 reasons for outliers. One, it's new science. A lot of great science has been based on outliers, points that stick out. You know, the classic one, penicillin. Penicillin worked because someone noticed that when he picked up his petri dishes, all his bacteria kept dying. It was an outlier. And he went, why is it always the mouldy ones that die? And he stopped and thought about it and discovered penicillin. So outliers are either that, or the other classic outlier is you just put the numbers in wrong. It happens all the time. Another way you can do it, so that one was just the points. Now I've got a box plot, which is another way of representing it. And all the box plot is it summarises information. So this line here is your average. Your box gives you an idea of spread. Also gives you your minimum point and your maximum point. There isn't one that's better than the other. It's what you're used to looking at and how you feel about it. And it's just a matter of practice and looking at it and going, does it make sense? So in this, to me, it looks like the horse has a slightly lower temperature than dogs and cats. I know that's true. That passes my stupidity test. I know the average temperature of a horse is 37.5, and the average temperature of the dog and the cat is about the same at 38.5. So these seem to make sense to me. Then I've got one now where, again, I've got box plots, but now I've got normal and disease. It says stupidity test. When you're diseased, you're more likely to have a fever. If you have a fever, you should have a high temperature. That seems to be the case. Here's the average temperature for normal. Here's the average temperature for disease. It definitely looks like there's a difference. I did this one because it was pretty. I quite liked it. I don't really have a good excuse for that, other than I thought it was quite pretty. It's a density plot. It's like a histogram, but a little bit prettier. One thing you do notice, though, if you look at the cat, notice you have two peaks. And I reckon that this is going to be your diseased cats with pyrexia, and this is your non-diseased cats. Now what I've done now is the same plot we had before. Remember I had the colours, but now I've coloured them by normal and disease. So you can see now, all the cats have a pretty small, they're about 38.5. The ones with fevers are a lot higher. You can see that the horse seems to be generally lower in the normal ones and lower in the diseased ones. And that really shows it up. You can definitely see the normal are all lower than the diseased, and the horse, a lot of variation in this horse one. A lot of variation in the horse, and one of the things I'll be asking is why is there so much variation, I wonder. And a lot of the time what I would do as a statistician is I would go and look at that and I go, oh, that's a lot of variation, why? And I go back to the researcher and say, is that what you expected? Is that fine? Is that what you wanted? Now you have the advantage that you are the researcher and the statistician. So normally when I do research, if it's in medicine, it's fine. I have a reasonable background in medicine, but I also do things in evolutionary biology, electoral roles, all sorts of stuff. And I spend a lot of my time going back to the researcher and saying, I found this, does it make sense? Because if it doesn't make sense, it's more likely that I'm wrong than they're wrong, unless I discover something new. So the whole thing of this is to see the patterns, see the things, does it make sense? That's an interaction plot. That just looks at the means. The main thing I'm looking for this is, it seems that the difference at this point between disease and normal for horses is more than dogs and cats, which is interesting. Perhaps horses are more likely to get a higher temperature if they get a disease state. You can also do descriptives. So here I've just done some summary tables. I did this one just to make sure that I knew that I had 10 results of each one. So this was just again me checking and going, when I typed in, did I have 10 horses, 10 cats, 10 dogs? Did I have 15 of each? And then I looked at this just to make sure my results I hadn't typed in something wrong and looked mainly at the maximum min and said, do they lie in the right place? And they do. And then finally I had a quick look at the average temperature, because I know the average temperature of dogs, cats and that. These should be about 38.5, which they're close to. This should be about 37.5. And this increase in temperature for fever is about right. Okay, so that's your look. And it's amazing how much stuff that you do will fit into that. And even though I put them in that order, I go, look, guess, decide, predict. It's very much an iterative process. I'll go and say, I looked at this, blah, blah, I'll do a t-test, I look at the t-test, I come back and say, oh, I did a t-test and saw this, does that make sense? No, it doesn't, right, I'll go back. I know we don't teach it like that. I know in ANOVA and stats courses, you're given a problem and they say, what's the test you would use for this? And you choose the test, and if you're right, you get the right one, and then you interpret it. That's all very sweet and lovely. That's not how I work. I work on two principles. First of all, model early, model often. So all the time I'm going, I'll try this model. Does it make sense? Can I improve it? Can I change it? Does it make sense? The other thing is, all models are wrong. Just some are useful. Simple as that. So I go back all the time and I'm looking and going, what does this model check me? Does it fit? Does it change? And I'll go back and do different tests. Start with t-test. The t-test worked. No, let's improve it. So now guess. So I want to get across an idea of what I'm trying to do here. So one of the key ideas in statistics is the idea that we've got, this is a stadium in Manchester. And let's have a thought experiment. Imagine that being the generous sort of person I am, I decide to buy everyone a coffee. So, and I'm only going to buy 2 coffees, so can I have a short black or a latte? So I go to each person and I ask them, do you want a short black and latte? Because once I know how many lattes and how many short black, I can put the order in. So a couple of ideas in stats is, first of all, we have this idea of what we call a population. So in this case, every person in that stadium is our population. It's everyone we care about. It's all the people we're interested in. So in your research projects, you might be looking at rats, might be your subjects. So now your population would be all possible rats that you're trying to model. Some of you will be looking at cell models. In that situation, you'll be looking at individual cells, and your population is all cells of that type. The problem is, I don't want to ask every single person what coffee they want. It's going to take me too long. In your situation, you want to get every single cell, I'm not going to get every single fibrosite and do the experiment on it. It's going to take too long. So what we do instead is we just take a representative sample. And the whole idea of the guessing is, I will look at my representative sample, and from that, I will guess what the population would have been. So I could go and select a load of people here and say, latte, espresso, get the proportion, and from that, I can now estimate the proportion of everyone in that stadium who would like a latte or a short black. So here's my very, very advanced diagram of this. The big orange cloud is your population you're interested in. So that's everything you're interested in. And that will have what we call parameters. And parameters are usually indicated by Greek letters. So you'll have something like the mean, which we represent often by mu, or it might be the variation of something in there, sigma squared. So I could be looking, for example, as how much variability do I have in red blood cells in the South Australian population? Or P for proportion. So these is my population, and what I do is I take a sample. And in my sample, I can now measure things like the sample mean, the sample variance, or the sample proportion. And these are the best guesses I have for those. So if I wanted to know, for example, the proportion of everyone who wanted a latte in that crowd, I could select 20 people, ask who wants a latte, and use that as my guess of how many people in total want lattes. So I thought I'd actually get an example. I've been teaching engineers. So I got 200 engineers and got them to email me their heights. So these are the heights of 200 engineers. And I actually know, this is my population. These 200 engineers are my population. And I know the true mean value. So what you could do is you could take a sample. So I've got 2 samples here. One sample are now coloured orange. And you can't see it very well, but there's grey samples as well. So I've got 2 samples. So the true mean of all them 200 engineers was 150.0415 centimetres. So that's the true answer. That's the answer that I wanted if I could just measure. In this case, I can. But if I take my sample, well, with the grey one, I got a sample mean of 153.57, which is my guess of this number. And you see, it's not completely right. It's close, but it's not right. And that's the problem. Every time you take a sample, you'll get an answer, and it will be close to the right answer, but it won't necessarily always be right. In fact, it will vary each time I do it. The orange sample, I've now got 148, so this one is actually less than the true value. And that's just because I want to know everything about these engineers, but I'm saving myself some time by just taking a small sample and using that to estimate the true value. The other thing you may have heard of is what we call a confidence interval, because sometimes that way you've got a single number. You take sample mean and go, look, I think the height of these 200 engineers is 100 and whatever. It's what we call a point estimate. So one-off number, and you go, here's my number, that's it. But sometimes you don't want just a number, you just want a range. You say, well, give me a range. What do you think, where do you think it lies between? And this is what we call a 95% confidence interval. So 95% confidence interval is a range. So there's a way of calculating it. I can go into the details later if you want, but basically, what I've got for the grey sample is I get a 95% confidence interval 148 to 158. So you're basically saying I'm 95% confident that the actual true average height of them 200 engineers lies somewhere between these 2 points. Basically, it's a guess, but it's a guess with a width. That actually contains the true value, and that actually contains the true value. In fact, you might ask what the 95% means. It basically means if I went and did a sample, so each of these lines is a sample. So I went and got a sample. This is a sample mean. This is my confidence interval. Next sample. Here's my sample mean. Here's my confidence. And I kept doing it. And the blue is the true answer. The blue is that 150 that we know is the true answer. And then I just said, when I did this confidence interval each time, did it actually hit the mark? Is the true answer in there or not? If it's in there, I coloured it black. If it's not, I coloured it red. And confidence intervals are designed so that on average, 95% of the time you get it right. 95% of the time, it actually contains the true value. So if you look there, there's 100. This case, 4 failed. 96 were okay. So that's all of estimation theory. We've done all of descriptive statistics. Now, we're going to talk about hypothesis testing. It's probably one of the most hated things ever. So, basically, this is how hypothesis testing works. In most Western countries, when you look at the law system, you are innocent until proven guilty. Okay? So if you think about the law sees it in 2 possible ways. You're either innocent or you're guilty. The entirety of hypothesis testing does the same thing. It has 2 choices. It's all about decision. You have 2 choices. You have one choice called the null and one choice called the alternative. Now for you, you'll often want something to show something of interest. So let's say you've got a new asthma treatment. You've got a new asthma treatment, and you'd like to show that it is a lot better at increasing your lung capacity compared with the old asthma treatment. So the thing that you want to do as researchers is you want to show there is a difference. So what you do is you assume there's no difference and then try and show that that's a load of rubbish. So in the law case, what you do is you assume anyone's innocent. What you try to do is say, are they guilty? So let's imagine that this is just a thought experiment, unless you turn on me, but let's imagine that someone comes in here and I've killed all of you. Just killed all of you, dead, as opposed to killed all of your life, which doesn't really make sense. So they come in, they find me, you're all dead, I have a knife, and I go, I did it, I did it, I did it. To be honest, they kept me away from my coffee and I was a little bit tense. So in this case, innocent until proven guilty. You have to go, well, we'll assume he's innocent. Let's look at the evidence. A lot of dead bodies, a knife, a lot of blood. Then you say, well, that's a lot of evidence. I'm going to reject that null hypothesis of innocent, and I'm going to conclude he's guilty as hell. Okay? So that idea is basically what we do in hypothesis testing. So what we do is we've got some data. So we've got this idea, we've got a null hypothesis and alternative. The null is the status quo, or what we're going to assume. The alternative is what you'd really like to show because that's why you're doing the research, but it's the opposite. So the null would be there's no treatment between the differences. The alternative is there's a difference. What we do is we've got all this data. You know, they found the dead bodies, there's the knife, the blood, the confession, the fact that I sent an email to Mark Hutchins saying I was going to do it. There's all that data. What we do is we have what we call a test statistic. And all a test statistic does is it basically takes all that data and shoves it down into a single number. And they're all designed, whether it's the t-test, the z-test, the f-test, the chi-square test, all of them are designed by statisticians such that when they get large, it's evidence that the alternative is the case you should go with. So all of them are designed such that, basically, if they're large, that's lots and lots of evidence that the null is a load of rubbish, so you reject it. If they're small, you retain the null. Now, just as a side point, it doesn't really matter, but we very rarely say the null is true. And this makes sense if you think about the law case. In the law case, If I didn't find you guilty, there's two possibilities. Either you were innocent, or we didn't find enough evidence to show you were guilty. Okay? So because of that, you can be proven guilty, but you can't be proven innocent. All we just say is, look, you could be innocent, or we didn't find enough evidence. So the same here with the null hypothesis. If our test statistic is small and we retain it, it could be that the null hypothesis is true. It could be, for example, there was no difference between these two treatments. Or it could just be you didn't find enough evidence. So because of that, never say the null is true. If you actually want to show the two things are the same, it's a completely different area in stats. And because I did my PhD on it, I would highly advise you to completely avoid it. It's horrendous. Yes?
I don't know if it's getting too technical, but is a test statistic just a difference? Is that what it is? So a large difference means you reject the null?
Usually a large difference, but we normalise it to take into account the variability as well.
Oh, okay.
So your T statistic, you look at the mean in one group, the mean in the other group, and then you divide it by how much that would vary.
Okay.
So you sort of take into account the differences and that variability as well. So the problem is, with the test statistic is, when is large, large? So we have a standard test statistic, which we call the p-value. And the p-value is the probability of seeing this data if the null was true. So basically, it will always lie between 0 and 1. And now, if your p-value is small, you reject the null. If it's large, you retain. So basically, it's the flip of that. Okay, p-values. Often you're told to look at 0.05. Does anyone know why you use 0.05?
Convention, isn't it?
Yeah, because we're convention and we're basically lazy. What happened is, a statistician called Fisher basically developed all of this. And he wrote, when he wrote this stuff down, he said, look, simple as this. In my particular experiment, he was looking at plants. I think I'm happy with about 0.05 as my cutoff point. But you need to make that decision based on your own experiment. Because we're all lazy, we went, forget that last bit, 0.05, that's a really good point. And we stuck at it ever since. What does it mean? It basically means at 0.05 that one chance in 20 you have of saying there's a difference when it doesn't actually exist. So in your research, if you're happy with that, if you're happy that you're going to sell your drug and say it's a lot better than that drug, but you can make a mistake when there's no real difference, about 1 in 20, use 5%. If you want 1 in 100, use 1%. If you want even more than that, take it into account. So that's hypothesis testing. Here's an example of one. There's lots and lots of test statistics, and I don't have a chance to do all of them, but you've got the general framework now. Basically, they're going to be large. What I've done here is I've got my data, so I've got my temperatures, and I'm going to do what we call a two-way ANOVA. So basically, if you're comparing two groups, use a t-test. If you're comparing 3 or more groups, use ANOVA. If there is more than one factor, so in this case we have the species and we have the disease state, then use a two-way ANOVA. If you had 3 factors, so you have disease state and perhaps whether it's a working horse or not, you have a 3-way ANOVA. This lady pointed out what the t-test does. All the t-test does is it looks at the difference in the means and basically looks how much that is, normalized for the variation. What ANOVA does is it's a slightly clever idea. Again, it was Fisher that did it. He went, if I looked at body temperatures is going to vary. If we took all our body temperature now, we'd get a lot of variability. Even if you took your same body temperature, from hour to hour, it would vary. So he said, you've got this variation. Let's partition it. So I've got this variation in body temperatures, and I'm going to start putting it in various buckets. I'm going to say, for this variation, I'm going to put a load of it in a bucket, which is the species bucket. How much of that variation is explained by species? How much is explained by disease state? And finally, how much is explained by the sort of disease state and the species? What I mean by that is perhaps when horses get fever, it goes up a lot more than when cats get fever. And all you do is you say, well, for example, if species makes no difference, the variability I get in this bucket, which is species bucket, and once I've started partitioning it, I'll always have a little bit left over. The bit that's left over, these two things will be about the same. That's all it does. So that's what this is doing. This is our output. You'd get the same in Prism. This is saying, here's my animal. This number here, the 1.9, is the amount of variation explained by species. This number here, the 0.4, is the amount of stuff that's left over after I've put all the variation in the species bucket, the disease bucket, and the interaction bucket. And then all it does is it looks at the ratio of this. It says, if these two numbers are the same, when I divide this by this, I should get 1. And that's all your F value is. It's measuring that. So this is a classic example. If your hypothesis is true, they'll be about the same, you get 1. As more and more the variation explained by species, then that number gets bigger and bigger away from 1. How far away from 1 is it? You just look at your p-value again. So this is for the bucket for animal, this is the bucket for disease state, this is the interaction, this is the stuff that's left over. And you can see, for example, here, this has a p-value of 0.607, so I would say there's no interaction. Get rid of it out of my model. So I do, there's your model. So there's a significant effect of animal on its body temperature, and there's a significant effect of disease state on the body temperature. Don't know where that is, but I know that there is a difference. And what you can do now is you can basically, once you've done your two-way ANOVA, which is taken into account, it's just asking the simple question, is there a difference? The question you want is, well, where is the difference? Now you can do your t-test. In fact, you can do in Prism also what we call pairwise comparisons. It basically says, now look, I'll compare each of these temperatures. I don't like the readout there, so all I did was, here's the plots. There's your 95% confidence intervals. So what's this showing? This is saying what I think the difference is between body temperatures of dog and cat. So there's your confidence interval. Here's your horse and cat, and here's your horse and dog. What you can definitely see here is that there's a big difference between horse and cat. Why do I say that? Because if this is the possible differences between a horse and a cat, notice 0 is not in it. So we know there's a difference, and basically, as I said right at the start, horses have temperatures lower than cats. They also have lower than dogs, but didn't pick up on this data, but such is life. So the last bit, predict. So to review, we've got descriptive statistics, which is look. We've then got our estimation theory, stuff like that, which is just a good guess. And then we've got a hypothesis testing, so you'll hopefully in your research do something like say, I want to compare this with this, off you go and do it. The final thing is the predict, and I thought the best way of looking at the predict is to actually have a model. So this is some data we got. This is for, I'm trying to remember, 624 students. So this was people who went from age 6 to 22, and what they've done is they've measured their lung capacity in FEV, forced expiratory volume, and their height. What they wanted to do with this data is they said, what we'd like to do is be able to go, have someone come into the clinic, look at their height, measure lung capacity, and say, is their lung capacity a lot less than we expect for someone of this height? Because as it is, we can use it as an early indicator of asthma. If their lung capacity is decreased, then we could argue that there's something going on that's they've got decreased lung capacity. So the whole idea of this data set was to make a predictive model. So when you go in, we can measure your height, we can measure lung capacity and say, we think we have problems here, you have a decreased lung capacity. So on here, we've got our scatter plot. Here is your lung capacity, measured in terms of logs, just to straighten up a little bit. And here's your height in inches. And so each dot here is an individual person. So this person here has a height of about 67 and a lung capacity of just above half a litre. And in this sort of situation, we have that, we would just do a linear regression. But then you can do prediction on your linear regression. So basically what I've done here is I've said which line fits this data the best, using what we call least squares. And so now I've got a line where I can predict. So if you come in and you're 55, I can look up here and say, look, I reckon your lung capacity should be about 0.6 litres. And again, you know how before we had our point estimate, and we said, here's your single point, here's your guess. You can also have an interval estimate. You actually do what we call prediction. So here what we've got now is we've got bands. So this is like your 95% confidence interval, but it's basically saying what I expect people to be. So now I can go up and say 55, your lung capacity should lie between this and this. If you're lower than that, we're concerned. We'd do some x-rays. Do further tests. So that was just an illustrative example that once you've got your model, you can use it to predict things. So, there's your stats framework in your 40 minutes or less. Look, guess, decide, predict. Most things will fit inside that. That's it. Done. Thank you. Cool. So you know, I have no idea what time it is. I've meant to talk 40 minutes and I almost got close to that. So, you've now got two choices. One, you could distract me and while I look away, you can run away like back and now I'm going to drink coffee. Or two, you have 20 minutes of my undivided attention. And I basically told you it's worth 20 minutes. So daft people pay you that. But so, any questions? Do you have research problems you'd like to ask? Is there anything I didn't explain? Is there anything I did explain by mistake?
Can you explain the difference between a repeated measures and a one-way ANOVA?
Yes. Oh, you want it as well?
Yeah.
Oh. It comes back to, I'll give you an example and I'll build from that. So, here's what I want you to all do tonight when you go home, because this is really cool. Basically, swearing increases your pain tolerance, which is really cool. So, what you're going to do tonight is you go home and you get a bucket of water, and you put ice in it, and then what you do is you put your hand in that water, you hold it in there as long as you can, and you time it. And one time you do it, you swear. Swear as loud as you want, and the second time you do it, don't swear. Say some non-sweary word like table. Right. And what you do is when you look at them, you'll find a difference. And this is what we call a matched pairs. So the idea of a matched pairs is that if I'm just comparing two groups, if I said to you lot, right, you lot are going to do the ice bucket with the swearing, because you look like a sweary lot, to be honest. While you, being very pious, are going to go for the non-swearing. What I'm comparing now is I'm comparing swearing and non-swearing, but I'm also comparing your pain tolerance with your pain tolerance. And the problem is, you might be a hardened bunch, and so you seem to last the water longer, you lot don't. So instead we can do a matched pairs. And so now, because you did both of them, you did the swearing and the non-swearing, what you're comparing is yourself with yourself. And so all that variability between individuals is basically reduced. Exactly what repeated measures is. So in one way or another, you've got your different groups, but you're often comparing, something that maybe is on one treatment with something that's not, and you're comparing the treatments, but you're also comparing individuals. Repeated measures has the nice thing that now you're taking into account the fact you're comparing each themselves. So in that case, you could actually have, say, 3 treatments, and you can put the people on and say the first time they get treatment A, then treatment B, and then treatment C. And then when you come to model it, you really don't care about the variability between individuals. You care more about the differences between the treatments. It actually can almost collapse down to the same thing pretty well. The only thing you've got to be very careful with repeated measures is, if you go back to the hand one, half of you should do it swearing first and half should do it non-swearing first. Because it may be that, for example, when you put your hand in the water, you become tolerated to pain. So if you do second, will always last longer anyway. So you'd randomise the treatment. So if you were doing a repeated measure to say treatment A, B, and C, I would have one group which would go A, B, and C, one which would go A, C, B, one group. And if you've got 3 things and you're going to look at all the things, you've got 3 factorial, you've got 6 possible combinations. Any other questions? Did it help at all? Or was it all stuff you've seen before, or was it just too much, too quickly?
No, that's great. It was well explained.
Good? I mean, the main thing I would say is, if you're not sure, come and see me. I can't teach you, I can teach you everything I know in about 5 minutes, but I can't teach you everything about stats in 5 minutes. But if in doubt, go and ask. I mean, the main thing I want to do is get to the point where you feel, I think there's a fear of statistics. I think people have been shouted at enough times by statisticians to be scared to have a go. Have a go, come and tell me what you've done. The worst I'll do is laugh. If you're lucky, I'll laugh with you. Maybe I'll probably point and laugh before other people point and laugh at you. But the idea is that it's just common sense. It's common sense with numbers, and if it doesn't make sense, don't worry about it. It is just, you're basically trying to prove it. You're trying to say, this is better than that. How do you know? Because here's my results, and here's how I summarise them. So do feel free to come and see me. Anything else?
What's the difference between parametric and non-parametric?
Good question. Basically, what happens is, a while back, say in Fisher's time, the problem is to do a lot of testing, you just didn't have computer power. So we developed a thing called parametric testing. What parametric testing does is says, if I'll make certain assumptions, I can make my calculations easy. So the classic one is, when you're comparing two treatment groups, if I've got my two treatment groups to go back and forth, if I'm willing to assume that the data is normally distributed, then I actually know that when it comes to calculate that p-value, I can calculate the p-value using what we call a t-table or a t-test. And it's basically saying, as long as I assume a parametric form, and by parametric form I assume the data follows a particular distribution, then I can get my answers out nice and easily. They're very, very powerful tests, but you had to make certain assumptions to use them. Because if them assumptions are not valid, they don't make any sense. So the other side of it, we had the whole non-parametrics. Non-parametric says, what happens if my data is not normal? I can't do the t-test, it falls down. So we always have what we call non-parametric versions of that, which basically say, I will make no assumptions, I won't assume normality and stuff like that. And what they often do is, the example would be the t-test. So with the t-test, I can have a load of people, let's say I have a treatment A and B, and I measure your lung capacity. And then I can do the two people and I can compare the average lung capacity of treatment A and treatment B. If I can't assume normality, what the non-parametric would say would be, John was in treatment A, but I don't think the treatment's making a difference. When I measured lung capacity and I got 4.5 litres, he would have had 4.5 litres anyway. It was just chance he ended up in group A. So if we did the experiment again, he ended up in group B, he would have still had that 4.5 litres. So in that case, you say the non-parametric approach is to say, I assume that the reason he's in any particular, it won't make any difference. So now you can number crunch, you can say to the computer, okay, you've got them results, repeat the experiment when Jono's in group B. Now repeat the experiment when us two are both in group B, both group A. And in fact, you can just say, keep doing it, and every time, let's look at the difference between them. Because the treatment made no only difference. So now what you're doing is you're creating what we call an empirical p-value. So instead of actually using your distribution to calculate that p-value, you say, let's run the experiment thousands and thousands of times and work out how many times we would have got this result, or as extreme, if it made no difference. So take home message, parametric is more powerful, you use it when you can, as long as the assumptions are valid. If they're not valid, there will always be an equivalent non-parametric, which is not as powerful, but because it doesn't make as many assumptions, you can use it nearly all the time. And you have, I can never remember them all. So t-test, the equivalent is your Kruskal-Wallis. There's also the Mann-Whitney, which are actually the same test. Basically worked on the great principle. Someone in chemistry invented this idea, said this is really cool, couldn't find it. And eventually he went, sorry, I'll just publish. Because you know if you publish and it's already been done, someone sent you a really rude letter. No one ever did. But then eventually discovered it was the same thing. So you have the Mann-Whitney and the Kruskal-Wallis, basically the non-parametric t-test. The non-parametric ANOVA is the Friedman test. And then sometimes you'll hear things like bootstrapping. I do a lot of, now we've got computers and I love coding. So I do a lot of simulations. I write the computer and let it. So I've done one recently for Paul Rowland, we're doing power calculations. And rather than do it analytically, I just run the experiment thousands of times and tell him how powerful this experiment would be. So, does that answer your question? Cool. Anything else? Right, I've recorded a lecture. I'll show it on YouTube for you. If you need to find me, I'm Jonah Chu, or talk to Mark, or Jonah, or Paul, or know where to find me. But please come and see me if you need me. Thank you very much. Cheers.